Welcome to my channel. In this session we will revise AS A-level biology for upcoming exam. Your exam is going to be commenced on October. So let's revise all the thing chapter wise through question answer through past year papers and answers. So let's get started. I have already made a video on cell structure and function. Now this session is discussion of past paper of molecular biology, the chapter 2. Okay, so the question here is, starch molecule are the main storage molecule in main types of cereal grains such as the grain of the barley plant. So you don't have to much focus on whatever the story is written, just focus on the starch. Okay, when the seed inside the barley grain germinate, gene coding of the for digestive enzymes are switched on. The enzymes that are synthesized catalyze the hydrolysis for storage molecules such as proteins and stuff. So don't focus on these stories, just focus on what it is just saying about the starch and the coding. Okay, so explain what is meant by gene. Gene is what? A gene is the small segment of the DNA that work for a special type of protein, that code for a special protein. So gene is a small fragment or small segment of DNA that code for a specific protein. Next is the hydrolysis of protein. In a barley seeds produce amino acid that can be used in the synthesis of proteins required for the formation of the seedling young plant. So figure 2.1 is an incomplete diagram of which molecular structure of the smallest amino acid glycine. So each molecule glycine had 2 hydrogen atoms. So you have to find out what is missing in this figure. So missing, we know that the formula is NH2 and C double bond OOH. Okay, so the formula is NH2, C here R is and H and C double bond, C double OH. That is carboxylic acid, amine group, and R is the functional, and H one hydrogen is there. And amino acid is also known as the substituted methyl group because it substitutes the H from here, H from here, H from here also. Next is starch is a mixture of 2 different molecules. Name the 2 molecules, that is amylose and amylopectin. Amylose and amylopectin. And you must know the structure of the amylose and amylopectin. Amylose is the single chain, whereas amylopectin is branched chain. Amylopectin is just complex and amylose is just a single linear chain. Next question is, 2 of the enzymes synthesized by the barley seeds are alpha amylase. The 2 enzymes we get from the barley seeds are alpha amylase and the maltase. These are involved in the hydrolysis of the stored starch during seedling formation. Okay, so in the food industry, you don't have to just focus on whatever all the things written and don't get scared. Okay. In the food industry, the starch extracted from the barley seeds is used for the production of the sugar syrup. Okay, so summarize the reaction catalyzed by alpha amylase. Alpha amylase is what? That the salivary amylase, or you can also call it as the ptyalin. So ptyalin what do? The ptyalin just break the starch into disaccharide, that is maltose. Here the barley starch is that converted into maltose, that is disaccharide. Okay, it is starch is polysaccharide and maltose is disaccharide. And how it happens? It happens by the action of the alpha amylase or the salivary amylase present on saliva. And then maltose is then converted into glucose by the help of maltase, which is present in intestine. So some of the substances shown in figure, it is given. Complete the table to identify which form in the term polysaccharide, which is monosaccharide and macromolecule. So you don't have to focus on all this story, just focus on which is in these, which is polysaccharide, which is monosaccharide. Okay, so glucose, we know that glucose is monosaccharide. Okay, glucose is monosaccharide. Maltase is just the enzyme, so it is macromolecule. Maltose is polysaccharide because it comes under disaccharide. So starch is also the polysaccharide. So starch is polysaccharide, maltose is also polysaccharide. We can say it is disaccharide. If the one option is also given disaccharide, then must tick on disaccharide. So when producing sugar syrup, there are advantages in using enzyme extract from the enzyme. For example, some enzymes. So don't focus on this. What the question is, suggest one other advantage of using enzyme obtained from microorganisms rather than enzymes extract from barley seeds. So you have to write the advantages of producing enzyme from the microorganism rather than the plant. Okay, so what are the advantages? First advantage is it is easy to culture. Okay, easy to culture, and it take very less time. In very less time, okay, less time, in very less time you get maximum amount of the microorganism. Okay, and they take very less time, they are easy to culture, and they produce multiple copies. So these are the advantages. Now third is the graph is given, and how the activity of the alpha amylase extracted from the barley seeds change as the temperature increase from 10 degree to 66 degree, 10 degree to 66 degrees Celsius. How it changes? We know that when we increase the temperature, first the enzymatic activity increase, and then at reaching the optimal temperature, it starts the enzymatic activity become deactivated, or it become very inactive. Okay, it become very slow. So explain the effect of temperature. So effect of temperature is, first it will get a start very rapidly, and then when it reaches to the optimal temperature, its activity will slow down, and at after a temperature it get denatured too. So sketch the curve, we have to make the sketch. And then question number 6, which row show a combination of molecule that could be found together in a single isolated virus? In a single isolated virus we get DNA. Yes, we get DNA, and we get protein too, DNA and protein. So this is the correct answer. We get DNA and protein. D is the correct answer. In most of the virus we have the RNA and with the protein, but RNA protein is not in the option. So many viruses also contain DNA as a genetic material. 7, a student carried out for 4 biochemical tests on the sample of the food. Okay, so emulsion he found test. Emulsion he found cloudy. Benedict test he found yellow. So Benedict test never gives yellow. Burette test found the purple color. Yes, Burette test gives the purple color, and iodine test give the blue-black color. So the answer will be burette. The protein is present. Burette test is for protein, so protein is present. Okay, and here fat is also present, but here what is written, fat is not present. Emulsion shows the fat. Okay, next is, which diagram represents a molecule of alpha glucose? So we must know, to have a alpha glucose, the carbon, in carbon 1 the hydrogen should be on top and hydroxide OH should be on lower side. So which diagram represents a molecule of alpha glucose? This one. Okay, A shows the alpha glucose. Next question, which statement about complementary base pairing are correct? Cytosine forms 2 hydrogen bond with guanine. This is incorrect, because we know that cytosine forms 3 hydrogen bond with the guanine. And purines and pyrimidines are different sizes. Yes, it is correct. Adenines form the same number of hydrogen with the thymine. Yes, as it does with the uracil, same. The base pair in a molecule of DNA are equal length, equal weight. Yes, so D is the correct answer. So guys, if you want to take the personal class from me, then you may ping me on comment box. What are the products when a DNA molecules replicate? What we get? We get two molecules of DNA each made of sequence of the paired nucleotides. So B is the answer. Now next question is a student carried out Benedict test on a sample and got a negative test. Okay, and got a negative test. So what would be the reason and how can he get confirmed that there is no sugar? So in such case he should have to boil that sample with the hydrochloric acid, neutralize with alkali, and then repeat the Benedict test. Okay, it may occur when the non-reducing sugars are present. Okay, the diagram shows three hexoses, 1, 2 and 3. Here is 3 hexoses is given. So what is the so 1 is what? 1 is sucrose. So sucrose structure is this one, 2. Okay, and the 3 one is the cellulose structure because it contains the beta. Okay, because in carbon 1 the OH is upside. If OH is upside, then it is known as the beta glucose, and if H is upside, then it is known as the alpha glucose. So this is the answer. And 1 is the glycogen. Glycogen is the stored glucose in the human body, that is present in muscle cell and in the liver cell. The diagram shows that the part of chemical structure of a natural occurring polysaccharide. So we have to name them. So this is the carbon 1 and OH is upside and H is downside, and here 1, 2, 3. Okay, so this is the structure of beta 1 linked with 3. So beta 1, 3 is written here. Beta 1, 3 and then beta 1, 4. Let's do this one also. Okay, so this is beta 1 and this is linked to beta 1, 2, 3, 4. So beta 1, 4. So this you can name the structure. This is very easy. What is the maximum number of water molecules produced when triglyceride is synthesized? It is 3. What occurs during the formation of peptide bond between two amino acids? When two amino acids get connected, then what happens? From the COOH, OH is removed, and from NH2, okay, one H is removed. Okay, like this. So water is removed. This reaction is known as the condensation reaction, and by this reaction they form the peptide bond. So OH is removed from the COH and H is removed from the NH2. In every case, whether it is the case of carbohydrate, whether it is the case of lipid, everywhere the condensation or hydrolysis reaction takes place. Condensation reaction is to join, is to connect to monomer, and hydrolysis reaction is to break the polysaccharides or the poly group into monomer. So next question is, this diagram shows the phospholipid molecules. This is very complex structure of the phospholipid given. I am giving you the simpler one. So this is the phospholipid structure, we know that. Okay. So this is the phospholipid structure. This is the phosphate group, which is the hydrophilic in nature. This is also hydrophilic. This is hydrophobic. This is hydrophobic. So this structure is just written like this. And the band one shows the trans 1 fatty acid. So what will be the answer? Increase permeability of the hydrophobic region. Which will increase the permeability of hydrophobic region? The Z one. Z will increase more the permeability of hydrophobic region, and repels the polar molecules. Which repels the polar molecules? It means water molecules. The Y and Z will repels the water molecules, and which will attract the polar molecule? X1. So the option is D. Next question. Some viruses have single standard DNA as their genetic material. The DNA molecule has to be folded to fit in protein code. So which statement about single standard DNA are correct? In some viruses, single standard DNA is present. So what is the correct statement? Single standard DNA cannot replicate semi-conservatively, And if the percentage of cytosine is known, the percentage of guanine can be calculated by the but the percentage of adenine thymine, this is quite wrong. If we know the cytosine, then we get the guanine, and if we know the adenine, then we get the thymine. But in single stranded, we must have the uracil in place of the thymine. And the hydrogen bonds may be present within the DNA molecules. Yes, this is correct and this is correct. Okay, so 1 and 3 will be the answer. Next question is, a piece of double stranded DNA is 12 into 10 raised to power 3 nucleotide is transcribed and translated to produce a single polypeptide. What is the maximum number of amino acid? Just divide it from the 3. Why? Because a codon is made up of 3. Okay, 1, 2 and 3. Okay, 3 nitrogen bases. So just divide it from the 3 and you will get 4. 4 into 10 raised to power 3. So this is the correct answer. Okay, next question is some codons are written here. Part of the nucleotide sequence of mRNA nucleus is shown, which is packed with spaces between the codon. So here some sequences are written and spaces are there. And what is the codon is there and amino acids names are there here. And you have to know, identify which one will delete or stop the coding or stop the reading, or which one will act as the termination of the polypeptide synthesis. So first is deletion of C from the leucodon. So where is the leucodon? This one. So if we delete the C, leucodon is CUA. If we delete the C and in place of C we know C always, cytosine always paired with the guanine. So GUA, no, GUA is not the stop codon. Stop codon is just the UAA, UAG and UGA. These 3 are the stop codon. So next one is deletion of C from TYR. Deletion of C from TYR. So UAC is written for TYR, means tryptophan and amino acid. If I delete the UA, then C will pair with G. So UAG is the stop codon. Yes, this will be the option. And the ribosome reaching the UAA. UAA is also the stop codon. So 2 and 3 will be the answer. 2 and 3 will be the answer. D is the answer. So I hope you like this video and you get all the answer. So if you want to study from me and if you want some classes and if you want more videos on it, please do like, share and subscribe my channel, and don't forget to hit the bell icon. Thanks for watching.